From Microbe TV, this is Matters Microbial, a podcast about the wonders of microbiology, microbiologists, and microbial centrism. This episode was recorded on May 8th, 2025. Hello, Micronauts, and welcome to our Quality Quorum for today. I'm your host, Dr. Mark Martin, Associate Professor of Biology at the University of Puget Sound in Tacoma, Washington. Today is the 91st episode of Matters Microbial. I think I, and Dr. Jennifer Quinn, should create some celebratory glow-in-the-dark pins to celebrate our 100th episode. I'll keep you posted. As always, I am so very grateful for your listens and views. As I've mentioned, the student chapter of the American Society for Microbiology at UT Austin invited me for a visit recently. As you can see from this flyer, they had many things for me to do. I enjoyed each one of them. At least I was able to bring my traveling tardigrade with me to represent Matters Microbial, as you can see. I had fun at my seminar, seen here, and met some wonderful students with even more wonderful questions. The LuxArt session was so much fun. Check out this giant pile of glowing Petri dishes. Makes me smile every time. One student got a little creative and made a spooky effect by moving a luminous plate around during photography. Awesome. Another student made a microbiologicals joke, seen here, suggesting that some microbes can invade biofilms. And I laughed out loud at the "pick me like an isolated colony" piece of LuxArt. I suspect that student had been doing a screening experiment in lab. Amy Baldwin, a student here at the University of Puget Sound, put together a 4-minute video of the LuxArt experience. Here is the title slide. I'll put a link to the video in the show notes. I hope you'll take a few minutes and enjoy the enlightening experience. Sorry about that. As always, the creativity of the students simply amazed me. I hope I can be invited to share my microbial sense of wonder at other institutions in the future, and I will always be grateful to UT Austin. In several prior episodes of Matters Microbial, we have explored the role that microbiomes can have on the organisms, including ourselves. But if you think about it, our own cells must fight invaders, promote microbial residence, deal with any number of nutritional and environmental challenges, tell friend from foe, and even shape whole microbial communities. The connection between macrobe, you and me, and microbe, within our gut, for example, is a two-way microbial street shaped by evolution, communication, regulation, and even chance events. So I am so very happy to welcome Dr. Emily Davenport of Penn State to the podcast to discuss the work of her research team exploring how human genes can impact the nature of our microbiomes. Welcome to the Quality Quorum, Emily. I am so happy to meet you.
Hey, Mark. I'm so happy to be here. Thanks for having me.
First, can I congratulate you on calling your research group Macrobes? You have a wonderful photograph seen here of your research group. Name that on your interesting website.
Thank you. Yes, we like all things macrobe and microbe here in the lab.
Well, it's all the same thing.
It is.
And a shout out
connected.
Yeah, and a shout out, of course, to Seth Bordenstein with the holobio concept that's everywhere.
Of course.
So I think a good place to start is you've mentioned to me that your work is primarily computational and associational. That's not a word, but you know what I mean. So that's important to keep in mind. But give people, the listeners and viewers, kind of a thumbnail sketch of what your approach is here.
Yeah. So in my lab, we're really interested, as you alluded to in your introduction, in human microbiomes. So I'm really fascinated as a human, as this kind of weird and crazy environment that all these microbes live in. In some ways, it's this ultra controlled, weird environment compared to other places on Earth, right? And so I'm really interested in understanding how, as a host, we control our microbial environments and how that affects our microbiomes. And to do that, we use primarily genomic approaches to ask the questions that we ask. So we're really interested in looking at all of the different members of microbial communities all over the body. And we're really interested in looking at the host side as well. So we use genomics of all the things here in the lab. We do microbiomes using high throughput sequencing genomics. And we also look at host genes and gene regulation as a way of how the microbiome interacts with the host.
You know, it's interesting when I teach microbiology, and I have a tiny snippet of one lecture to deal with the human microbiome, right? Awful of me, but just one microbiology class at a small liberal arts institution. And so I like to talk about, and I'll put a link in the show notes, to Sarkis Mazamian's original work showing the different sides of things. In other words, alter a particular host gene and you can kick out a member of the intestinal microbiome. Or you can change something about that organism and it isn't able to colonize. So it's that two-way street.
Absolutely. Yeah, and it's so much more, that is true, but it's also so much more nuanced than that. Because the fact is, you alter a gene and you kick out a microbe. The fact is that doesn't happen usually in people, which is a good thing, right? Like we're not walking around getting bombarded with things that give us mutations every day, except for the sun and other cancerous things. But it's all these tiny little differences between people that can potentially affect our microbiomes. And understanding what those differences are and how they affect those microbiomes, I think, is super important for understanding what makes a quote unquote healthy microbiome. I think it's really important to understand, number one, the kind of baseline, how does this system work? And then, of course, in the context of human health, we have to understand what's happening when things are healthy to understand what happens when they're diseased and how to fix it. So I think you're totally right. It's hugely important to understand that kind of give and take between both of these two things.
I'm glad to hear you say that, because so much of science in the biological sciences has been reductionist for so long. One gene, one phenotype, that kind of thing. And nowadays with systems biology, you can look at patterns. So if I gave an incorrect statement about Sarkis's work, that wasn't my intention. Because we don't do knockouts in people to figure out what's going on, at least yet. So the whole point is, hopefully never. It takes us back to Jurassic Park, doesn't it?
A little bit, yeah.
But what I would say is that I often tell people that we're like a long tube. In fact, our friend in common, Jill Waters, actually drew that on a cup I have in my office. We're just a long tube, and it's true. And to me, it's interesting to think about what enters that tube and what exits, what holds them in place. And I think that you have, as you've mentioned before, the co-evolved residents, which is fascinating. We have transients that are just passing through, if you'll forgive me, or forgive this even more, squatters that might set up shop temporarily. So this whole, and as I think you've alluded to as well, one end of the tube is very different from the other. So it is a really unusual community. And I'm reminded of an essay by Ed Yong, There Is No One Microbiome. And I'm curious what you think about that.
It's 100% too. I think, so first of all, I would say, complicated things versus reductionist things, we need all of it in science, right? I'm 100% a believer in sometimes you got to start with the really complicated things, look for some patterns, look to see what's interesting. And I definitely fall into that camp more so. I'm very much a pattern finder. That being said, to understand truly how a system works, you need to take a reductionist approach too. You need to start with something really simple, build it up, knock things out, see what happens. So I really believe the best science happens when you have both things happening and playing off of each other. And I think you're also totally right in that we are just one big long tube, but it's a complicated tube, right? The tube, number one, it's really long. It's really twisty. It's really turny. It has different textures. It has different inputs depending on where you are in the tube. And even if you took, even if you put yourself through a sectioner and you took just one little tiny section of the tube, even within that section, there's variability, right? What's happening in the center of your digestive system is very different than what's happening at the edge of the digestive system, right? And so, and again, this goes back to it's just so complicated. We need to take a big view and understand kind of broadly what's going on. And then we need to dig into each of those little tiny communities and figure out how that's interacting with the host or interacting with each other, or, how long is this going through the gut, or is this kind of hanging around because it's, clamping onto host mucins or whatnot, right? So there's all sorts of different questions you can ask depending on where you are in the gut. And they should be asked because they're all very interesting.
Well, if you'll forgive me saying it's not a big tent in science, it's a big petri dish for our purposes with lots of different approaches. And I really do think that's important. I am concerned the way that microbiology is presented to the public a lot. And that's why I'm delighted that you're here to talk about kind of the complexities of things. How many times have we heard and again, I don't mean to be disrespectful to anybody, if I eat this yogurt, I won't be depressed anymore. And I mean, we see this everywhere. I call these angel microbes as opposed to the devil microbes that always do terrible things. And it's not like that. We're a series of ecological niches and, along that tube, as you say. And one of the most important things for listeners and viewers to remember, and I know you believe this way too, is not necessarily to believe hype, even scientific hype.
It's hard because I think so many people are so enthusiastic about what we do or the potential of what we do. But there is that backside of, if let's say you are, selling somebody some probiotic yogurt that doesn't actually do something, then you kind of burn bridges with the public, right? It's a very delicate balance to make. You want to be enthusiastic. You want to, express the potential of a technology or of a field, but without overselling some snake oil. Yeah.
So taking a moment to listeners and viewers, it's interesting to think, let's just assume for a second there was a microbiome associated with people, but there are going to be differences from person to person for a variety of reasons. Could you expand upon that?
There are huge differences between people and their microbiomes. And I think that's actually something that if you are not in the field, maybe is underappreciated, because one of the things many people learn is the stat about your own genomes, for example, right? Like most of our genetic content is the same. We only differ in a few little tiny things spread across our genome. And that obviously affects us. We look different, we sound different, we do different things, we have different interests, all of that stuff that has a genetic component is there. Our microbiomes are wildly different from each other. If you get a group of individuals in a room, chances are they're going to only share, some maybe 20% of the microbial species, and they're all going to be in very different abundances across individuals. So there's huge differences. There's no one thing as a healthy microbiome. It probably is something very personal depending on your physiology, your behaviors, your actions. And all of those things affect your microbial community. So the diet you eat, obviously, if we're thinking about the gut, the medications you take, your exposures, your activity levels, your immune system, and of course, one of the things I'm really interested in is host genetics. Depending on your host genetics, that may, confer a different microbiome configuration than the person sitting next to you. So all of those things kind of feed into creating a different microbial community in your gut and other places on your body.
Yeah, and I'm really glad you said the latter part in particular. We have this tendency to think that I remember reading that there's a different microbiome associated with the right hand than the left hand, and all of that is really cool work. And maybe you can say that, but going from person to person, there's going to be all that variation that you're talking about. And as you say, I'm sure temporally there might be differences throughout the day or from week to week. Many of us have probably seen that. I guess it was Rob Knight who did it many years ago, the trajectory of the gut microbiome of an infant.
Mm-hmm, mm-hmm.
I'll put a link to that in the show notes. But that's just one individual.
Yep. And Ruth had a really great study as well, tracking her kid over time and just how things change. And it's fascinating. And there are some trends, right? Like we know that things kind of with infants, for example, they mature around age 3-ish, but there's huge variability, right? Like, what is matured for one infant is different from another kid. And there's differences across countries and there's, we can talk about broad trends, but there's huge amounts of variability, and understanding why that variability is there. Does it matter? And can we learn from that? I think is hugely important.
Which leads us to your own interest, which is the role of host genetics. That is to say, our own genetic makeup in that business of who's there in the microbiome.
Yep, exactly. Yeah, so I got started in the microbiome field, maybe 15 years or so ago. It's when microbiome stuff was really taking off. It was like, the late 2010-ish area. So we were really starting to sequence a lot and learning a lot about human microbiomes. People were studying diet, antibiotics, all of those things that are obvious major drivers of gut microbiomes. One thing that hadn't really been done yet is people looking at whether or not your host genetics can influence your microbiome. And it's kind of logical that it could, right? Like your host genetics controls a lot about your physiology, including, pH of your gut, your immune system, all sorts of different things. And so maybe there could be a role of your genetics in determining what microbes live in your gut and at what abundance. So I'm really interested in coming at it from that angle because I kind of view humans as like this little weird vessel that carries a bunch of microbes around. And those genetic variants control that environment in your vessel. What metabolites are you giving to your gut? What immune system functions are you encoding? How strong is that immune system? How tight are your barriers in your gut? All of those things can influence the microbes that you have in your gut. And so I really wanted to understand that from a genetic perspective to see, okay, theoretically this is possible, but do we actually find evidence that there are microbes that are heritable, genetically heritable? And can we identify what host genes, pathways, functions actually control those microbes in that environment? So that's what I've been doing for the last 15 years.
So some of the things technologically you're doing that we've discussed on the podcast before, but one of the things that I'm curious about when you mention abundances, how are you measuring abundances of microbes in a given sample?
And I should specify, we are only measuring relative abundances in humans. So most of my studies, we get a piece of poop from one of our participants. We extract the DNA, we sequence that. And what we get is basically a snapshot of what the microbial community looks like in that time. But we're only sequencing what we sequence. And with humans, it's really hard, if not impossible, to actually measure absolute abundances. Like, how do you measure how many microbes are in that giant tube that we just talked about? It's impossible. And so we do the best that we can to look at relative abundances and see if host genetic variation is influencing that.
And you're doing this with molecular techniques. It's important, I think, to remind listeners and viewers, I don't know what percentage of what lives in the gut can be grown in the lab, but it's small.
Yep. And even if we could, each individual has hundreds of species that live in your gut. And so it would take a very long time and a lot of manpower and a lot of energy to try to sequence all those things. There's value to doing that, and there are certainly people working on that, and that's great. But if we want to learn about populations, which is what I'm interested in, you can't do that for hundreds or thousands of individuals, at least not within a lifetime.
This reminds me very much of trying to establish a marker for human genetic diseases. And there's all this challenge with it because there's a lot of diversity in human genetics.
Yep, exactly. And so, I get to have this problem on both sides of the coin where, in human genetics, of course, there's a bunch of variation that you can measure and that's pretty common across individuals. There's also a bunch of variation that's very unique to you or to me. And it's really hard to look at those variants and figure out what they're doing because they might only exist once or twice on Earth. How do you study that? The same is true in the microbiome, and in some ways the microbiome is a little bit worse because our microbiomes are so different from one another. Like, you just need a lot of samples to be able to look at things that are even common and have power to do that kind of thing. So yes, the variability is both wonderful. That is what we are studying. That is what evolution works on. That is what makes us different and unique and interesting. It also makes our jobs really challenging because it just means we need to look at more and more and more.
By the way, I have a quote, and I'm going to have to put it up. There's a scientist I know at UC Riverside who once talked about an organism as a big bag of other organisms walking around.
It's true.
Yeah, it is. So how do you deal with this? And you have a particular model system humans that you're looking at.
Yes, my model system is humans. I don't only look at models; I look at all of us as well. So in humans, we do a couple of things. Like, I really am interested in studying actual human samples from actual humans because I think if we really want to understand what is happening in a human community, you just have to look directly at the community. And then once we kind of sort of figure out what's going on, I think, like we were talking about earlier, then you can take a reductionist approach and like really dig in and tease out individual mechanisms and that kind of thing. So for some of my past work, I've done what's called a genome-wide association study. This is where we look at human variants all across the genome and try to identify ones that are associated with the microbes that live in your gut. So to do this, we take fecal samples from people and we take some of their genetic material, and then we can do associations between all of those different things. It's a ton of different tests, and we need hundreds, thousands, ideally more than that samples, but they're hard to collect and expensive. In the lab right now, we've moved towards doing integrating host gene expression analysis with microbiome data. And the idea here is, number one, we're focusing directly on mucosal microbiomes now rather than fecal microbiomes. The idea being that, right? I know, I'm really excited about it. The idea being that, like, if we think the host is interfacing with the microbiome, that's probably happening at that mucosal barrier. And so by studying those mucosal microbes, which is a different subset than the luminal microbes, we might see more of those effects. And by focusing on gene expression, this is giving us a little bit more biologically interpretable results. We can actually identify genes and host pathways and that kind of stuff that's associated with those microbes and try to glean some insights into the physiology that kind of links those two things. That's really challenging to do from GWAS alone.
I have to tell you that in the minor work that I do at this institution, because it's an undergraduate institution, I have a collaboration with a colleague, and we study the microbes that live within the cloacae of lizards. I can tell you about it another time. But it's actually really interesting. And what was funny is I kept insisting, what are you measuring when you look at fecal samples? Now, I understand this is controversial, but I forced people to do this experiment. You get very different results in something that's 2 hours old versus freshly deposited. You're getting a bloom, in other words, in that fecal sample. So I am beyond delighted if you're studying the intestinal microbiome that you're looking at mucosally associated microbes. Thank you.
Oh, yeah, absolutely. Well, and I think especially for what I do, it makes a lot of sense, right? Like, fecal samples are mostly luminal microbiome, mostly determined by diet. And so if you're studying diet, great, study fecal samples. That's fantastic. If you're studying host genetics, it's, the signals that we get from fecal samples are really small. Does that mean that the effect of host genetics is small? Maybe. Or it just means we're not studying the right microbiome. And so I'm hoping by moving to mucosal samples that we're getting a little bit more direct of a picture of the things that are actually modulated by the host a little bit more directly.
It makes me so happy. Thank you.
Me too. Thanks.
All right. So, differences between individuals. So, and you mentioned a little bit of your GWAS, as it's called, to find possible genes associated with particular types of microbes. Can I ask a question that's a little bit off topic just for a second?
Absolutely.
Do you have a favorite member? I'm hinting, a favorite member of the gut microbiota?
I have multiple favorites. I love them all equally. But I do have some that I hold in high esteem. We were chatting about this a little bit earlier. I love Akkermansia. They're fascinating. There does seem to be a genetic component to Akkermansia, which I can talk more about. Bifidobacterium as well are one that pops up again and again in genetic studies. So I also have a soft spot for Bifidobacteria.
So will you talk at the moment? I mean, I want to put some links up because most people, when they hear about the gut microbiome, they'll do things like say, oh, that's mostly Bacteroides, even though that's a different taxon now, I know, which I have trouble enough with the English language. Imagine what fun I have with that. Yeah. But I do want people to know about Akkermansia and Bifidobacter. So
definitely. Yeah. So in our GWAS studies, which we've done in multiple populations, and these things pop up multiple times, so they've been replicated. Basically, what we find are that there are variants in the human genome that are associated with the abundances or the relative abundances of these taxa. So depending on if you have an A, T, G, or C at a certain location in your genome, right, you might have more or less of these bacteria. And why is that? So the Akkermansia story is not quite as clear cut as the Bifidobacteria story, but there are variants that lie outside of a gene that has been associated with obesity. And this, when I saw that, I was really, really interested because in the literature, Akkermansia has been shown to be protective against obesity in both humans and mice. So the more Akkermansia you have in your gut, the leaner you tend to be. Mouse experiments where they've actually put Akkermansia into a mouse has shown that it is causal, like it is protective. So the fact that we're seeing genetic variants in humans that lie outside of a gene that's been associated with a body mass index in humans, and this connection to Akkermansia, which has shown a causal role in body mass index in humans and mice, I thought was really, really interesting. That's the extent of the story there so far. It's a little bit hand wavy. We don't exactly know what's going on. The fact is the human variant that we're detecting, it is not in a gene. It is outside of a human gene, which means it's probably regulatory in nature, but we just don't know that much about it yet. So we'll see. The other bacteria that's super interesting is bifidobacteria. And this bacteria has been associated. Oh, do you have a little plushie of one somewhere back there?
I do.
In your plushie microbiome? Awesome. Well, the cool thing that we see with bifidobacterium is that the variant in your genome that actually confers lactase persistence is the one that is associated with bifidobacterium. So individuals who are lactase non-persistent, so if you're lactose intolerant, those individuals tend to have more bifidobacterium in their guts, which when we first saw this was a little counterintuitive. Right? So bifidobacterium is one of these bacteria that is highly prevalent in infants, and it tends to kind of decrease as we age. We know that bifidobacteria are really good at utilizing a bunch of compounds in milk. So milk oligosaccharides, a bunch of other milk sugars, and including lactose, they're able to utilize as carbon sources. So right away when we saw this association, we were super excited about it. But yeah, then when we actually broke it down by genotype class, we were pretty surprised to see that it was the lactose intolerant individuals that had more of this, potentially milk utilizing bacteria in their gut. So what's going on there? And what we thought was going on was that actually in individuals that are lactose tolerant, you drink a glass of milk, that milk passes through your stomach into your small intestine. You express lactase, that digests down the lactose, and then by the time that food reaches your large intestine, where the bifidobacteria live, there's no more lactose carbon for them to eat, so they're pretty low abundance. If you're lactose intolerant, you drink a glass of milk, the milk passes through your stomach, into your small intestine, you're not digesting the lactose anymore, so then that lactose makes it to your large intestine, the bifidobacteria see it, they use it as a carbon source, they flourish, they grow to higher abundance. So that was kind of like the story we came up with, but we didn't actually have the data to test it at the time. Subsequently, there have been other people that have had sufficient data, and they see exactly this prediction. So in individuals who report drinking milk, we see this association with bifidobacteria. In individuals who report not drinking milk, this association completely goes away. So it's really a gene by environment interaction that affects how much bifidobacteria you have in your gut. So it's a super cool example, I think, of this interplay between diet and host genetics and how that might affect your microbiome.
I'll dig up some references and put them in the show notes. That's a wonderful story. Thank you.
Yeah, there's a great schematic that was in a review paper that we wrote that kind of outlines this hypothesis about, milk drinkers versus non-milk drinkers. So I'll make sure we get that added into the folder.
You have that wonderful review paper about the host. Is it in the host genetics review paper that you did?
Yep.
That is a wonderful, wonderful article.
Yeah, great. I'm glad. People should read it. It's great. It's a really cool subject, I think.
We've talked a little bit about the technology. We've talked about the association of host genetics with a little bit with the relative abundances of certainly of bifidobacteria, and maybe Akkermansia, and a nice discussion of Akkermansia. Can you talk about some other projects you're working on?
Yeah, so we in the lab right now, we've got things going on in a few different areas. We're certainly following up on some of these genetic things. Like I mentioned, we're really interested in looking at interactions between gene expression and the microbiome as a way of kind of teasing apart a little bit more what these stories are between associations on the genetic side with the microbiome. So I just told you about bifidobacteria and Akkermansia, and we have some kind of hand-wavy stories around each of them. We can at least kind of theorize a mechanism that might be why host genetics is controlling these bugs. However, for most of the GWAS hits that we've gotten, we have no idea what they do. They're variants that are intergenic in humans. We have no idea what gene they regulate. We don't know anything about them. We don't know what tissues they're acting in. We don't know anything. And so we're doing a few things to try to figure out what those associations might be. Number one is looking at gene expression. You might think, if thinking about central dogma, right, you go from host genetic variant to gene expression to protein abundance to your ultimate phenotype. There's more arrows in between. But right, so if we kind of move one step closer on the dogma to the ultimate phenotype, maybe that will give us some more insights into like what are the actual physiological mechanisms that link host genetics to microbial abundance in the gut. We are doing this in human samples. So we've got some samples from gut, and we're also looking in the lung at these kind of gene expression microbiome relationships. We keep talking about kind of maximalist versus minimalist approaches. I love doing things in humans, but the fact is we're very limited as to what we can do, right? We can find associations and then we're kind of stuck. So we're supplementing that with some in vitro co-culture work in the lab as well. So we're actually growing up human cells on a dish, exposing them to different microbes, and then looking at how gene expression, chromatin accessibility, all sorts of different things happen, how they change on the host side of things to try to tease apart these mechanisms. Are there certain metabolites that microbes are producing that regulate host gene expression? How exactly is the host responding to these things? So the kind of view there is we kind of learn some things from human samples, then we take it into the lab and we try to dissect it out with these, kind of crude techniques that are only an okay proxy for a human being.
So are you using, can they make organoids of intestinal cells?
They certainly can. We are not using that system only because it's very, very finicky. So we might move there in the future, but right now we are just working with, Keiko-2 cells, things like that.
I think that this is a wonderful idea because while it's possible, as I alluded to earlier, to make specific knockouts in, say, mice, if you have a gene you think is associated with, I'm just going to say friend or foe. I know that's not what you mean. But, if you think about it, how do you maintain your friends and how you exclude your foes become very, very interesting. And there must be a host set of host genes involved with that, as you allude to. And at least in a mouse system, you can study it. And certainly you can do knockouts in human cells. So it's a great way to get like some footholds in the work.
Exactly. And especially for things where you just need a lot of material. Even in a mouse, mice are small. And so, and expensive and hard to make. And, making a genetic knockout in a mouse is also not trivial, right? So, I think there are benefits to all of these different systems, and I'm sure we'll use many of them over time.
I had a previous podcast guest who was talking about using human cells in culture to test certain antivirus medications because of the interferon response. And this is what it reminds me of to some degree, that you can use it as a proxy that works pretty well.
Exactly. And especially the types of things that we are studying in the human cells are molecular traits, right? We're studying gene expression. We're studying chromatin accessibility in those host cells. So we don't need a whole organismal response. If we needed a whole organismal response, obviously we would want to do it in a different system. But the fact that we are studying specifically molecular phenotypes means that we can use something like a cell model, and hopefully that will be like good enough that will replicate what happens in an actual like full organism.
And with all due respect to other model systems, I really like you looking at the human cells as your proxy rather than, please forgive me, my good friend Caenorhabditis or Drosophila or fill in the blank.
So we do also have Drosophila happening in the lab.
I have questions. Go ahead.
So like I said, right, we like doing stuff in humans, but humans are hard to study. We like doing stuff in cells in a dish, but the fact is, like I just said, those cells aren't great for organismal phenotypes. I'm really interested in organismal phenotypes related to the microbiome. So one of the things that I'm really, really interested in is this idea that we've evolved with our microbiomes and that they might play roles in fitness-related traits in a host. But the fact is, studying fitness is incredibly challenging or impossible in humans. And even in mice, it's long, it's hard, expensive. And so one of the things we are getting into in the lab right now is doing some work in Drosophila. And I love the Drosophila system for this because, number one, you can rear them really quickly to large numbers, and they're very, very easy to make not abiotic. So all you have to do is kind of bleach off the outer part of their eggshell, after the eggs are laid, and then as long as you keep them sterile, then you can inoculate them with whatever microbial community you want and then study the effects of that community on that organism. So we're doing way too many things in the lab, but basically we're trying to study really complex systems, simple systems, and then also a system that has more of an organismal phenotype component to it.
So my friend Nicole Broderick isolated this Serratia relative and a little bit smelly, as some Serratia are. And I understand Germans don't pronounce it that way, but I will. Anyway, the point is, I call it the Red Death. One of the problems with doing any kind of pathogenesis work in Drosophila is to get the pathogen into Drosophila. This is lethal orally.
Mm.
And so when they eat sugar water that I've spiked with the Red Death, when they die of it, their bodies turn red. That's why I call it the Red Death, right? And so I don't really work on that very much, but the point is there are all these tools available for Drosophila work, and it's been shown many times that how you study, for example, disease progression in Drosophila can relate to what you see in mammals. So it isn't weird science, and it's really important right now that we tell listeners and viewers, the public in general, that sometimes the science that seems not directly attributable to humans or applicable to humans actually is.
Absolutely. There's so much cellular machinery, the way that our cells operate, the way that our organs operate, the feedback systems between different organs within an organism. That there's so much of that is shared between humans and Drosophila. I mean,
Yes.
I'm not one to put humans above all others. I think all organisms are interesting and all biology is interesting. That being said, if we want to make a public health argument, it's totally valid to study something like Drosophila. You can just do so much more and so much more quickly, scanning things and trying to figure out the system that you could always then later bring it into mice or humans or something later.
Absolutely. And so I'm really excited by this work that you're working with Drosophila. I will say that I've never had much luck surface sterilizing the way that you do, because they're inside
finicky.
baseball. And they're very hydrophobic. And so I would look inside my tube and there'd be a little bubble around the Drosophila, right?
There has been extensive troubleshooting of this in the lab, because the fact is also different Drosophila lines have different needs. So this is also one of the fun things about this project as well, is this is something that is undergrad led at the moment, which I find really rewarding. I'm sure you appreciate that. And so it's been one of the fun things for the undergrads to kind of like work through and figure out a new protocol. Like, okay, now we've got a new Drosophila. Wait, this isn't working. Oh no, okay, now we got to go back and figure out how to do these wash steps. And okay, this one needs it a little bit harsher and this one needs it a little bit lighter. And, there's so much of science that's art. And I feel like that's one of those areas where it's just like, tinkering the recipe a little bit to get the best, tray of cookies.
I wish that we could have like members of the public come in and just do an experiment once in a while.
Yeah.
Because I really, and you will appreciate this. I have a friend, Amy Chang Volmer, who used to make her administrators, where she was a professor at Swarthmore for many years, come in and do an experiment so they understood what it took.
I think that would be super helpful, especially because the image that you get of science in movies and TV is so not how it actually works, right? It's slow and it's methodical and it's not glamorous, except for the very rare instances you have like a breakthrough, right? And I think kind of, giving people that experience just to see, oh yeah, no, it takes a long time. It's hard. Most things don't work the first time you try it, right? And it's just kind of a slog, but it's a slog that's worth it in the end.
So I really like the research areas that you've been in, because it takes things that we're kind of narcissistic, so we're interested in human related stuff, of course. And though you can't do experiments on humans, and that's a good thing, you can do them on cells, versus the microbiome issues, and trying to kind of tease apart those sorts of things. When I teach about Paneth cells, for example, that's a lovely topic to bring up with students when they realize their recognition. Margaret McFall-Nye insists I call them MAMPs, for microbially associated molecular patterns. But those things that our own cells recognize that say, this is a bacterium or this is a virus, you can see it's valuable to fight off something that might potentially cause disease, but it's also helpful in recognizing the things that we've co-evolved with.
Mm-hmm. And I think there's a growing recognition among everyone right now that we shouldn't just think of microbes as germs, right? We grow up with microbes, they help train our immune system. And if you overdo it, that results in issues as well. And so I think that's been something that's been really nice, is that there is an appreciation of the fact that, like, it's a complicated system. And it's not just that our bodies
No.
want to shut out all bacteria.
No.
Really, we need to kind of figure out which ones are the ones that are going to harm us and which are the ones that, might be useful to keep around because they help digest our food and they help break down our drugs and they help do all sorts of things that kind of keep us healthy, right? And so I think it's a really fun time to be a microbiologist because I do think that there is that appreciation out there.
Well, I mean, one of the first buttons that I made for my students, first evolved, last extinct, which is what Abigail Salyers, rest her soul, and I came up with many years ago. I even have this on my arm, which in bad Latin says, "All hail the small masters." And I think it's really important because they were here first and they'll be here after we're done. And the fact that some of them actually are helpful to us, but most don't really care, is an interesting subject. And the ones who don't care fascinate me because they're passing through, as we've mentioned, all the time.
They've got
So,
their own things going on, right? Like
yeah,
they're living in the world. They've got their own stressors, and they're trying to survive off of their carbon sources and fight their predation and do all the things that they're doing. And we're just their weird obstacle, right? We live in a microbial soup, right? We can't see it, so I think it's underappreciated, right? But microbes are everywhere. They're on every surface. They're floating in the air. They're everywhere, and all of those
yes,
microbes are, affecting our environment and the world that we live in. We've never lived in a time without microbes, right? We've
right.
always had them doing whatever they are doing. And we've grown up and evolved and all of that in that environment. So yeah, I think we have to appreciate what they're doing all around us.
Oh, absolutely. I mean, I want to proselytize on this. I want to be a microbial evangelist. And to let you know ahead of time for fall semester, last time I'm teaching microbiology before I retire, which is different than expiring. What I have for the logo is, it's a microbial world. We just live in it. After the Frank Sinatra story, right? It's Frank's world. We just live in it. And it's really true. And I'm really glad to hear this aspect of it. But again, when we talk about our interactions with the microbial world, we have to be able to identify those that we could care less about. We have to identify those that could potentially be a problem and those that we've co-evolved with, that friend or foe. Maybe friend, foe, or indifferent are the signals that we need.
Maybe.
And that's where I think so much of the value of your work comes from, I think.
Well, thank you. Can I ask you a question?
Anything
I'm
you like.
curious, as someone who is, as you say, nearing retirement, and you've been teaching microbiology for so many years, have you sensed a shift in perception about the microbiome from your students? Like, do you feel like, 10, 15 years ago, they felt differently about it than they do today?
Yeah, I think you're right, and I'm glad you brought this up. When I first started talking about it, it was like very difficult to get them to appreciate that there wasn't one microbiome, and it didn't matter that that's the same in any organism you choose to look at, and how you tease out the ones that are incidental from the ones that are co-evolved. And that's why I talk a little bit about some of the knocking out of various friend or foe functions. I wish I had a more eloquent way of putting it. And I find nowadays the current climate is people are looking for the microbiome to do things, either positive or negative. Arturo Casadevall does not like the term dysbiosis, for example. And I think I don't know him. Professor Alm at Harvard feels this way as well. And the issue here is that when you say something is out of balance, that kind
Implies a
of infers
balance.
implies
Yeah.
a balance. And I think if we're going to say that, it's like ecology. There's more than one balancing point
Yeah, totally.
because it is ecology.
Yeah. Just little ecology. Yeah. Hmm.
Yeah,
Yeah,
but
I tend to agree with them. Yeah.
yeah, and I think it's interesting because nowadays I see things. I'm not going to name them. You've seen lots of things on the market being presented as good for your microbiome now. That's new.
Yeah.
And to the point where I seriously want to get some diagnostic primers and see what's there. Because
Yeah.
I don't know that it's really what they say. I hate saying that, but it's true. I mean, for example, I'm waiting for someone to suggest Akkermansia as a supplement. Have people been trying to do that?
I honestly, I don't follow because I think probably similar to you, that all just kind of, I have no interest in that. So I don't know. Probably somebody out there has tried. The thing I always wonder is like, even if you say it's what it is in whatever product you're making, does it actually get anywhere that it's going to do something? Like how much survives past the stomach? I don't know. And if you don't keep taking it, is it really going to colonize, or is it something that will just be transient? I like the fact that there's potential in that. And I'm sure that there are some products out there that, really do work for some people. But it's hard to know if something is going to work in anyone and if that anyone is going to be you, right?
Without naming names, there's apparently a company that sells an anti-hangover microbiome. Have you seen this?
I have not.
I will send you a link. Anyway, I like talking about that with students, and I'm so delighted when they bring up the fact of how do you know it got there? And again, maybe those studies have been done by a company. I don't know. I do know for some probiotics, people have done that. And as you say, there's so much diversity along the tube, right? And I'm not just even talking about stomach acid. There are other things that have to be, and the competition with what's there. I remember people, and you remember this too, they said the microbial world is all about like getting along versus a Marshall viewpoint. But I kind of think that microbes are constantly jostling for position, sometimes literally.
It's like any other ecological community, right? There's competition, there's cooperation, there's different niches, there's, all of those things are at play all of the time.
No, I agree completely.
It's a constantly dynamic ecosystem.
So I'm happy to have received that question, and that's my answer to it. But I have a couple of things I'd like to chat with you about. May I?
Absolutely.
Other than the work you discussed showing a relationship between lactose tolerance and intolerance in the microbiome, do you have any other examples of direct up or down regulation having to do with the microbiome?
Yeah, so there have been a handful of studies that have directly looked at gene expression in the gut, for example, and the microbial communities directly adjacent. And we see a number of examples of different genes that seem to be associated with microbial composition. I'm not thinking of any of the names off the top of my head, but those studies are out there for sure. I can drop some links if the listeners are interested.
Certainly, I think that'd be fun for people to see. Do you have any thoughts about the microbiome and drug effectiveness? We've discussed this on some previous episodes of the podcast.
Yeah, that's a really interesting area. It's not one I study. So I will say that, up front, it's not my area of expertise. There's some really, really great people like Peter Turnbaugh and his group that are working on this. Lisa Meyer over in Europe have some really interesting studies. There's definitely been examples, right, where the community of your microbiome and the functional capacity of your microbiome has been demonstrated to interact with drugs, like whether that be, break them down faster or slower, or make them so that they are working in different ways. Like both of those examples have existed. So yes, this is definitely an area where there's a lot of interest because obviously, we give people doses of drugs and we don't take into account what their microbiomes might be doing at all. And for the most part, maybe it's just like little differences between people here or there and how a drug is utilized or whatever, but there might be certain circumstances where there is an interaction that's a little bit more extreme. And we want to understand those cases and figure out how to take that into account in clinical care.
I think I've seen some indications that this can even impact how chemotherapeutic drugs work in cancer treatment.
Yep.
And again, with what you're saying, with the variation between individuals, this might be, obviously it is an important area to study.
Especially in such a critical point of life, right? When it's such an extreme treatment, and oftentimes, cancer patients are, more prone to infections, or they've been in a hospital environment and those types of things where your microbiome also, might be not whatever your quote unquote normal microbiome looks like, right? So understanding kind of what those interactions are super important for understanding how to treat those patients better.
One of the things that we've been very interested in the podcast is how microbial communities interact. I mean, it's one thing to think about host versus microbe, but interactions between the communities that exist, is there any way that can be addressed via, I don't know, computational studies, AI?
Good question. So certainly people have been interested in this question of like, do the members of a certain microbiome interact potentially? There's a bunch of, network methodology that exists out there. So you can take existing sequencing data and you can try to predict, whenever we see this microbe, this other microbe is also really high abundance. Or whenever I see this microbe, this other microbe goes away, right? And it's not perfect because you don't know why that's happening. It could be cooperation, or it could be that they're actually in two totally different niches and they have nothing to do with each other. We don't know, right? Will AI be the answer to that? Maybe. The thing with AI, right, is that AI, you have to train AI models using a whole bunch of data. It learns from that data. It picks up patterns that are, for us, incredibly difficult to see. And then it applies them to new data to learn new insights. That works really well for things like text, where there's, billions and trillions and gazillions of text bits out there on the internet for AI models to learn from. With microbiome data, it's much more sparse, right? Microbiome data is really expensive and difficult to collect. So I think there's huge potential there, but we just need to generate more data to act as a training data set so that we can get some of these AI tools working better for microbial questions. I think there's potential there, but I don't know that we're there yet in terms of that technology.
And we're not even thinking, once again, if I trivialize this, I don't mean to, the geography of the tube.
Yeah. And oftentimes, at least in human studies, we can't even really take that into account, right? All we get is a fecal sample at the end, which is this weird amalgamation of everything from that tube. What is that, right? How do we actually, disentangle that into the parts of the different parts of the digestive system? It's really a difficult problem.
I was looking over some of your publications, and you did some stuff that I thought was really fascinating. Would you care to talk about ancient dental calculus?
I'm happy to. I will say I'm not lead author on that publication. I just pitched in a little bit. That was work done by my colleague Laura Wyrick here at Penn State. If you haven't had her on yet, she'd be a great person to talk to. Very interesting. But so she does a lot of work looking at ancient microbiome work. She focuses on dental calculus because it's one of the best places to get ancient microbes. You can imagine, there are ancient fecal samples. They're called coprolites. They do not preserve very well, as you can imagine. That though, the dental calculus, oh, you've got some, good, of course you do. But dental calculus preserves the microbes really, really well. Like there's a biofilm that forms on your teeth. Those microbes, even after an individual passes away, they're kind of protected from the elements because they're in that biofilm. And so even with samples that are hundreds of years old, thousands of years old, she can go in, actually scrape off that calculus and study the microbes in those individuals. And so she's done a lot of really, really cool work. The paper I was associated with was looking at a bunch of oral microbiomes in Britain across time spans. And we see that there is a huge shift during the Industrial Revolution, which is probably not unexpected, right? Food sources changed, hygiene practices changed, density of people in cities changed. There's so many things that changed with the Industrial Revolution. It's not very surprising that it, is apparent in our oral microbiomes. But Laura's also done really fascinating stuff with like Neanderthal microbiomes and things like that.
Oh, I've got to learn more about that.
You should talk to her. She's really interesting.
Will you put in a good word for me?
I absolutely will.
So that's one thing. And then I noticed that you had some association with twin studies and the Hutterites.
Yes. Yeah. So all of my genetic work has been done in those populations. So when I was a grad student, I worked with the Hutterites. So for the listeners who aren't familiar with the Hutterites, which I assume is most of you, because I was not familiar with them at all before I started working with them. They are a religious isolate group that lives in the Western United States and Canada. So they live in South Dakota, Montana, and then upwards into Canada. And the deal with the Hutterites is that they were this kind of persecuted religious isolate group that kind of shifted around Europe for a whole bunch of time. And then about 150 years ago or so, a group of them came over and settled in the U.S. And it was a pretty small group. It was only a few hundred that started. And now the Hutterite population is something like 30 to 40,000. And it's a pretty isolated community. They live communally in these little towns called colonies. And they do use modern kind of modern lifestyle things. They use electricity. The kids sneak in cell phones, all that kind of stuff. So you can kind of think of them like Amish, but they're not like really Amish, but they are a religious isolate group. And what makes them so fascinating to study is that they do live and eat communally. So every day for 3 meals a day, the women in the colony go to these huge industrial scale kitchens. They make these huge meals and they set them out buffet style. The entire town comes and eats all 3 meals all together 3 times a day, every single day of the week, their whole lives. So if we're thinking about studying the microbiome and reducing dietary variation, it's a really fascinating population to study because basically everybody's on like a super controlled diet, right? And of course there's personal choice. Like it's a buffet style thing. If you don't want to eat the lima beans, you don't have to eat the lima beans. But right, there's kind of a limited variety, among individuals at any one time of what they're eating. So as someone who's interested in genetic effects, this was a really interesting population to study because these dietary effects were minimized compared to kind of other populations in the US. So yeah, so I looked at the Hutterites and did genome-wide association studies in that group. Another great thing about Hutterites is we have extensive details about their family histories. So we have the full, family trees, and we can take that into account in our family studies as well. And so this is the study where I found things for Akkermansia, for example. We started getting those first hints there, and Bifidobacterium as well. Then as a postdoc, I started working with twins samples. And the great thing about twins is that there are multiple types of twins. There's monozygotic twins that share exact genome copies, and there's dizygotic twins. These are twins where they're technically from 2 separate eggs, so they have the same genetic relatedness as any other pair of siblings. But they, were, gestated at the same time, and you have the same kind of exposures as you were developing. And so it's a really powerful system because you can compare these monozygotic twins to dizygotic twins, and this can give you an estimate of the genetic component to whatever trait you want to look at. And of course, the trait I'm interested in is microbes. And so we used these pairs of twins to estimate actual heritability of microbes living in the guts of these individuals. And what we found is generally across the board, for common microbes, most microbes are not heritable. We see no signal of heritability. These are microbes from stool. Keep that in mind. However, there were some microbes that showed heritability estimates of something like 30%. So, things like height in human populations has a heritability of 80%. So that can kind of give you a little metric of, they're not the most heritable things in the world, but there's measurable heritability. And this, again, is pointing to the fact that our host genome is doing things, that controls the abundance of microbes that are in your gut.
This is so super because when I used to teach genetics and people would come up with the idea, can we find a gene associated with a particular genetic difference? I would talk a little bit about this kind of work, and I would talk about twin studies. I would talk about small communities like the Icelandic populations prior to about 1940, the LDS communities where they keep superb records, and the Hutterite communities. And as I mentioned to you before, when I was in college, I wrote a term paper on the Hutterites. So
yeah, and it's, this is a microbial podcast, so I won't go too much into the human genetic side of things. I wear 2 hats. So I am a microbiologist, but my PhD is in human genetics. So I come at things from kind of both angles. I think both things are fascinating. But one of the things on the genetic side of the coin is that, I think, it's easy to learn about genetics in that framework that you mentioned where, there's a gene that does X. For most traits in humans and other organisms, it doesn't work like that, right? Most things are very complex. The height that you are isn't because you have a tall gene, right? It's the effects of many, many variants all across the genome, and all in combination is what ends up, getting you to the height that you are, along with your diet and all the other environmental exposures and whatnot. And we think the same is true for microbes, right? It's a complex trait. It's not just one gene that controls the amount of Akkermansia that's in your gut. It's probably a bunch of different variants that are acting in a bunch of different tissues all over the body that control things like the pH of your gut, or the way that you metabolize your diet, or your immune system function, or the composition of your mucus in your gut. All of those different things, right, are all in combination, contributing to this different environment that these microbes are living in. And so it's really identifying what all of these little different individual things are and how they work together that influence the microbial community in your gut and elsewhere.
Oh, no. I mean, I used, again, when I taught genetics, I would talk about this, that we instructors lie to you, making things seem much simpler than they are. And even Mendel himself was playing fast and loose. And I'm not making fun of that. I'm not being negative about it. It should be more complicated, because if it wasn't that complicated, we'd be in a lot of trouble, I think.
Absolutely. And it's how you have to learn as well. And same thing with microbiology. You teach the simple things first to get that foundation, and then you go back in and you're like, actually, everything is a mess. Everything is complicated. There's always an exception to the rule, right?
Yes.
You know, these perfect operon systems that you learn about in bacterial genetics, they exist. And then there's all these other way more complicated ways that microbes regulate their gene expression and how they respond to physiological stressors, right? And that's the beauty of, I think, all science, is that, you know, you do what you can, you learn what you can, but really it's such a messy and complicated world we live in. And it's going to keep us employed for a long time, hopefully.
Well,
Maybe not you. You're going to retire.
Wow.
Peace out.
Peace out. Listen to you. All right. Well, you know, I can I just have a few words as we start to kind of slide down to the end of the podcast about your journey. You mentioned that your training was in human genetics, which is very interesting to me. I used to think I wanted to be a genetic counselor, and so I took counseling classes so I can use that voice if you'd prefer. But my point I'm trying to get at is I really love the genetics part of it, and then I mixed that with bacteria to get where I am today.
Yeah, I have a very direct path for where I ended up. I have always loved microbes. I will say I'm a microbiologist first and foremost. My very cheesy college essay reason for loving microbes, and it is totally true, is that I took a biology class in high school where we streaked out plates, and just the idea that you made the invisible world visible was something that was truly just like, it caught me. There was something about that I was like, I want to study these things that are normally invisible, that control all of this stuff. So even from the time I was in high school, I knew I loved microbiology. My undergraduate major was bacteriology. Again, loved microbes. When I was in college, it was pre-next-gen sequencing days. So everybody had like their favorite bug, and they were studying their favorite gene and their favorite bug. And that was cool, but I didn't have that. I didn't have a favorite bug. I didn't necessarily have a favorite gene that I loved. So I knew I loved doing research, but I just wasn't sure exactly what I wanted to do. So I ended up working for a couple of years after graduating, and I landed randomly at a genomics company. So I worked for this company called Roche Nimblegen, and I worked doing sequence capture for next-gen sequencing, and I fell in love with all things genomics there. I loved seeing patterns in data. I loved being able to look at the entire genome and see what's weird, what's normal, what is this, what is that, how do things change in different populations. I also, when I was at the company, I was a tech, I was a pipetter monkey, and all of my friends, though, were on the bioinformatics side of things. So they were all coding, they were all designing the arrays, they were working with scientists to design the experiments. And I looked at their jobs and I was like, that's really cool. I want to learn how to do that. So I knew after a year or two, I was like, okay, I'm going to go back to grad school. I know I want to do genomics, and I know I want to learn how to code. And in 2008 or whenever this was, I thought the only way I could do that is by studying human genetics. Everybody who's doing genomics is studying humans, right? So I went to grad school for human genetics. And then at the end of my rotation in the lab that I ended up joining, I was having a chat with my PI, and he was like, well, you can do genomics of microbes. It's the microbiome. And I was like, I have to do that. Like, that's absolutely what I have to do. And so I've been really lucky that we basically, I was able to come into a project where I could study the genomics of all the things, right? I could study the genomics of humans by studying human genetic variation, and the genomics of the microbe by studying the full microbial community in those individuals, and really combining those into one. So yeah, it was a circular path to get there, but I started as a microbiologist, detoured as a human geneticist, but ultimately I get to do both things, and I've loved it every step of the way. And I get to code, and I get to do wet work, and I get to do all the things. It's great.
Your pipetter monkey thing makes me laugh, because I have a theory at some companies there's someone banging a drum as people are pipetting from tube to tube. Maybe it's that way. I will say my experience in what I tell my students, my experience in industry was a wonderful one. I joke that I was a pipetter monkey, but I worked at a really great company, and I had a lot of ownership over what I did, and I learned a lot, and it was very scientifically enriching. So I actually went to grad school thinking I would go back into industry, but I fell in love with teaching, and here I am. I'm soothed stuffing pipette boxes. Okay, I don't know about you, but
I don't miss that. I'm not going to lie.
or when I have a lot of plates to strike. I love it. And one of the advantages of being at a small institution is that I'm in the lab every day.
Yeah.
And you're actually working the lab too. But a lot of people at RO1s write grants. They have to be written.
Oh, I do not get into the lab anymore. I will be the first to admit I'm losing my skills. Hopefully it's like riding a bike and it'll come back to me if needed. But no, I'm definitely behind the computer way more than
as long as somebody doesn't bar entry to your own laboratory, because I've seen that happen.
Yeah.
Well, I have to thank you, Emily, for this wonderful opportunity to chat with you about things. And the most important thing that I want to say is that you were able to give some insights onto the host side of the human microbiome. And I really appreciated your enthusiasm and your humor. And
well, thanks, Mark, for having me on. It's been a real pleasure. I always love chatting microbiome, anytime.
Well, again, I wish you and yours the very best.
Thanks, you too.
This has been Matters Microbial, a weekly podcast about the wonders of our microbial world and the people who study it. You can send questions, suggestions, or comments to me at mattersmicrobial@gmail.com. Show notes from today's episode, with epic links as always, can be found at microbe.tv/mm. If you like our work, please consider supporting us at microbe.tv/contribute. I'm Doc Martin, and you can find me in the biology department of the University of Puget Sound in Tacoma, Washington. Dr. Emily Davenport is in the Department of Biology and Huck Institutes of the Life Sciences at Pennsylvania State University in University Park, Pennsylvania. Many thanks, as always, to David Renata for superb editing and Reber Clark for the wonderfully quirky music. I hope that you've all enjoyed being part of our Quality Quorum today. See you next time on Matters Microbial.